Really.
Hello, everyone. Nice to have you here. We'll wait for 1 more minute and get started. Ooh, is Yinka here? Can't find him. He promised to lead the discussions today, but I can't find him. See if he's away, actually. Okay, let's see if he joins in. Otherwise, is someone willing to lead the discussions if he's not available? Basically, what you have to do is to just talk about the tasks, explain your understanding of the topic. You could just pick one, or you could talk about more than one. Explain your understanding of the topic. Specify points where you don't understand or you are unsure about. And if there is something you think you know and you want to enlighten others about, specifically about the topics, you can point it out as well. And of course, you can also ask for clarifications, and others in the meeting can also ask for clarifications generally. We just need someone. I don't want to do all the talking. It feels boring that way. Let it be a dialogue, you know? Um, okay. Nobody wants to lead the conversation. Okay. Wow. Okay. If nobody wants to lead, I would go ahead.
I have a question regarding the task 2.1.
Uh, First we try to explain the task. Then if you have a question, you can bring it up afterwards.
Yeah, I mean, we were given some metadata and data as two files. The metadata has some information like about the strains used. There are three strains, strain 1, 2, and 3. And each of the strain has like a wild type one and a mutant variant. And there are two repetitions or replicates of each strain experiment. I didn't understand that part, what the rep means. And I have like a data where we have OD values versus time.
Okay,
And
so we'll get to those points. Like I
yeah,
said, first we want to give an explanation of the task, and if you have a question, you can bring up the question after the explanation of the task. Okay.
just a second. Yeah, the task says plot all the growth curves of OD 600 versus time for
So do
different
you want to
strains.
explain the task first?
Yeah, to explain the task, I have to read it out, right? That's what I'm saying.
Do you want to go ahead and take charge? It's just a question. Do you want?
Yeah, I'm just telling like I'm telling my understanding about the task.
Ah, okay, then
Yeah,
go ahead.
yeah. So I have to plot like growth curves
Mm-hmm.
of OD600 versus time for the different strains given in the data set, right?
Mm-hmm.
And it also asks for a growth curve for like a knockout and knock-in strain overlaid on top of each other. Is that like wild type and mutant? That's
Mm.
my guess.
Hold on. Uh, okay, so first of all, you are jumping to your own question. Don't jump the gun.
Mm-hmm.
First of all, you have to plot OD600 against time. Do you know what this means? Do you know what OD600 means?
Yeah,
What is OD600?
OD600 is like the measurement of turbidity. It's an
Okay.
indirect measure of the number of cells.
Mhm.
Mm.
So when you measure OD600 against time, what do you have? When you plot it against each other, what do you have usually?
We get a curve depending on the number of cells in the solution or whatever.
So you get a growth curve, that's what I mean.
Yeah.
You
Mm.
get a growth curve.
Mm.
So for each strain, plot a growth curve of the knockout and the knock-in strain overlaid on top of each other. Um,
Mm-hmm.
and you are so the knockout. So basically, so someone asked us to share my screen. So the point is, we believe everybody should, but I can share my screen. My opinion is that everybody has access, but I remember not everybody is. I can stop sharing for now. I would share the Slack interface specifically. Da, da, da. Yeah. Yeah. So I believe you can see my screen.
Yes.
So,
Mm.
so let's quickly, so I don't want to, I have to share my entire screen. Sorry. I will share my entire screen instead. Okay. I guess everybody can see my screen now. So you have the data sets. Um, this is the entire data set, right? It contains time. So because we asked for the plot of time against OD600, so this is the time values, and you plot against OD600. Then for each string, plot a value of plot a growth curve of the knockout and the knock-in. So the knockout will be the mutant. So for example, let's look at the description of the data set as well. So we have a description of the data set here. So we have the strain. So I think knocking should not actually be wild type, but in this case, we consider it as wild type because the knocking is overexpression, if I understand. The knocking should be overexpression, but in this case, we just use the wild type as our knocking strength. Okay, so the strain replicates one is A1 for the wild type, is A2 for the mutants, A1 again for the wild type, A2 for the mutant. So there are, let me understand again. So the strain information, the wild type, the mutant, intro, so that, yeah, so people don't get confused. Okay. So we have the strain, we have the wild type. So for the wild type, multiple strains, we have A1, A3, A5, A7, A9, and A11. So basically, if you want to plot the wild type, so you should understand that this is not represented well because this is just a text viewer. So this strain is here, and you have information about each strain. And for the wild types, you have information about them. The mutant is A2, A4, A6, A8. So basically, you get information about all of them. And they are just increasing repetitions of each other. That is multiple replicates. So typically, it's assumed that this kind of experiment was done in a high throughput setup. That is, you did multiple screens at the same time. You screen multiple things at the same time. So you can have multiple replicates of the same thing. You can have like 10 to 20 replicates. But in of the same, so there's something you call biological replicates. So I would consider this rep one and rep two the biological replicates. And for example, if you have wild type occurring once and another wild type occurring again, I'll call them technical replicates of the same thing. So basically, you should be having the same thing with this wild type and this wild type. The experimental setup, we don't want you to go too deep into that because experimental setups vary from experiment to experiment. But in this case, just consider this as wild type and consider this as mutant. So the wild type would be your knocking and um the what's it called? The mutants would be your knockouts. So continue. Who was speaking? Mabindu. Or you are done?
Yeah. Yeah. I mean, so we have three strains. Am I correct?
Mm, yeah, three strings.
So for strain one, the wild types are A1 and A3, and mutants are like A2 and A4. Is that correct?
Yeah.
So in
So it's not just A1 and A3, it's A1, A3,
mm,
B1,
yeah,
B1, B3,
B3.
C1, C3. So it's that
Yeah.
way.
So in that case, like if I have to plot growth curve for strain one, like which shows mutant and wild type,
Mhm.
should I take an average of wild type values and then mutant values and make it less like two curves?
Yeah, this is up to you. Some
Okay.
people like to
Mm,
plot average. Some people like to plot the individual plots one by one.
okay.
Yeah.
Anything is fine, right? Okay.
Yeah, yeah, yeah. So it depends on, you can also read papers and see what people do.
Uh-huh.
But first, I would say you should plot the individuals first. If
Mm-hmm.
you realize that there's not so much difference, maybe you
Mm-hmm.
can just plot the average. But I like seeing the individual plots first
Okay.
because mean kinds of like, so it's like pulling a lot of samples together and it makes people appreciate your work less somehow. Just my personal opinion. It doesn't mean it's correct. So the thing is, I did a lot of work and you want me to just show you one. Sorry, no.
Good.
You see up to 12 plots. So basically, this is just my opinion, right? It doesn't really count. Like, I mean, it doesn't really mean this is the standard, but depending on how you want to interpret, if you want people to see the variations in your data, I think it's good to plot all of the plots. Like we have one, two, three, four, five, six graphs. It's not too much. But
Mm-hmm.
if you just want to pass a single message, no, you can just plot your mean or pooled data.
Got it. Got it.
Okay, let's
Yeah.
continue. So using your function from last stage, determine the time to reach the carrying capacity for each strain slash mutant. Do you remember this, Imabindu? We're having a
Yeah.
conversation.
Yes. Yeah. Yeah.
You have to mute.
Yeah. No,
uh,
I was actually stuck at the first part itself, like the data and everything. The rest
okay.
seems like okay, I could go through them, but first I
Yeah,
have
yeah,
to
we have to go
understand the
through
data,
everything now.
right? Yeah.
The point of this meeting is we are going to
Uh,
a meeting. So and you said you want to discuss this with me.
yeah.
So we are discussing it together. Don't go mute on me again, please.
Sure. Sure. Okay.
So generate a scatter plot of the time it takes to reach the carrying capacity for the knockout and the knock-in strains. So do you understand this question?
So, like, we have to apply the logistic function to this data and like make
Hmm. Don't
just
get.
note down that I mean, like, point out the time takes to reach the carrying capacity in case of like each strain.
Do you remember we calculated how to reach the carrying capacity from last stage?
Yes. Yes.
How did you do that?
I used a logistic function which involves the initial population size,
No, no, no,
the
no, no.
growth rate,
Yeah,
and so, like pass some parameters into the function.
I think you're mixing it up. So what we want to calculate is the carrying capacity, not the um, not the generation of the logistic function itself.
Mm-hmm.
So you are to write a function for determining the time to reach 80% of the maximum growth. So basically, we are not, we already have the plot. We already have the curve rather.
Mm-hmm.
What you have to do now is to calculate 80% of the maximum
Mm-hmm.
OD.
Mm.
You did this right in the last stage.
Yes, yes, yeah.
80% of the maximum OD and
Mm.
use and find the time at that point of
Mm.
maximum OD. And probably you solved that through your logistic function, which is possible. But I actually wanted you guys to calculate just 80%, 0.8 multiplied by the maximum growth, and then
Mm-hmm.
determine the time for getting
Mm.
to that point.
Yeah.
And this is the
Okay.
carrying capacity.
Mm-hmm.
Now, what's happening? My Slack disappeared. Okay, hold on. I don't want us to spend more than 15 minutes on a question. But yeah. So you now have 6 growth curves for wild type and
Mm-hmm.
another 6 growth curves for the mutants, right?
Mm-hmm.
So the 6 points from the um, mutants, you calculate the carrying capacity. So you have 6 values for carrying capacities.
Mm-hmm.
Then you have another 6 values for carrying capacities of the mutants. So you have
Mutants.
for the wild type and you have for the mutants, and then you plot it against each other on a scatter plot.
Ah, got
Yeah.
it.
Or another idea might be to generate a box plot.
Mm-hmm.
So I think, yeah, okay. It's actually there also. So you could also think scatter plot or box plot, which is better to actually represent this kind of data. It's a very
Mm.
good question. And
Okay.
now, is there a statistical difference in the time it takes the knockout strain to reach the maximum carrying capacity? You know, is there a statistical significance? So you have to so basically you have to do something like what test would you do in this case? For me, I could try t-test.
Just yeah.
Yeah, I
Mm.
could try t-test, and with a t-test, I could see I could just collect all the carrying capacity values. Basically, by the time I'm talking about carrying capacity, I'm talking about the time it takes to reach the stationary phase.
Okay.
I'm talking about the time it will reach stationary phase. So you just take all the values for what's it called? All the values for the mutants, you compare with all the values for the uh,
Wild type. Yeah.
wild type, and then you generate a
Mm.
p-value. Depending on that, you make your observations as comments in your code.
Got
Was
it.
that helpful?
Yeah.
I probably took all the discussion from you. I'm sorry. Yeah, because I wanted you guys to lead the discussions. Because this is the most interesting part of bioinformatics, talking about projects. Okay, that's okay. I think I have some questions. Let me quickly go over it. What is OD 600? You should have known this from the last stage. Yeah, it's a measure of turbidity and indirectly detects the number of cells. Yeah, but you could also read this online. Yeah, Yinka said, yeah, Yinka is finally. Can I ask something about data? Do we have to have to take the average of OD 600 values for each biological strain, for each biological replicate for? So it's up to you. You can decide to uh, have pooled values, that is average, and you can decide to have singular values. It's entirely up to you. Just communicate it while you are reporting. I think that's the most important thing. In science, there is sometimes no accurate or no correct way. Okay, so as long as you can communicate the exact plot or the exact graph you generated, I think it's fine. I hope I answered your question, Milton. Please, why do we have 6 graphs? Because we have, yeah, we have 3 replicates, but we have two technical replicates as well. So we have three biological replicates from the strain level. Then we have two technical replicates, that is from the same culture on that day. For example, if they did, so I hope you're what is it called? You're a microbiologist. But okay, what I'm saying is from the same batch of culture or overnight culture. I don't know whether it's still. I'm a microbiologist, so I can talk about this. But if I'm trying to, I don't know your discipline, but if you can tell me, I can probably find it something relatable. But from the same batch of culture, can you? So people do exactly the same thing within the same batch of culture. Then they do two different batches of culture. So those two are the two different batches of culture are biological replicates. Let me see if I can. I don't know how to describe this well. I think it's too technical. Let's look for an empty screen. So let's say, for example, this is a, oh my God, this is my conical flask. And then I have a culture here. This is another conical flask. Let's assume these. Yeah, this is another conical flask. And I can assure you that you won't get any conical flask that looks like this in the market. Um, so from this conical flask, you take you can take this cell and use it for your you can basically you can put everything on plate, and then if you, for example, if you're using a petri dish, you can plate everything. So these two are biological are technical replicates of each other. But again, let me change to red. So you have cells here. So you have a technical replicate here and another technical replicate here. So this is one, this is two. I know it's a lot of abstraction. It's very difficult to follow, but just think of it as an experiment, and you have two replicates. Now, within each replicate, you still have multiple replicates of the same thing. So then you have, but this is a biological replicate one and biological replicate two. And this is technical replicate one, technical replicate two, technical replicate one, technical replicate two. So you have biological replicate one, technical replicate one, biological replicate one, technical replicate two. You have biological replicate two, technical replicate one, and biological replicate two, technical replicate two. Yeah, science can be that uninteresting. I'm sorry. Yeah, I'll clear my screen. Okay, Melton ask a question.
Hi, Wally. Could you go back to the screen with the metadata? So
Mhm.
based on your example here,
Mhm.
say for strain one,
Mhm.
rep and rep two,
Mhm.
A1 and A3 are technical replicates, and rep one and rep two for strain one are biological replicates. Is that?
No, no, no. I would say, yeah. So this is strain one, strain one.
Yes.
They have the same replicate. Sorry, they have different replicate numbers. So I'll call this A1 and A3 are biological replicate one. So this will be biological replicate one.
Mm-hmm.
And B1 will be biological replicate two.
Mm-hmm.
No, 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 sorry. Sorry. This is
The
biological
definition of
replicate.
biological and technical replicate is kind of
Yeah, it's quite confusing, but it's the only explanation I have to to tell you that they are all the same, but
Mhm.
they're just sampled at different under different wells or under different plates or under different conical flasks.
So they're all all
It's the
samples.
same set of conditions. You just have four replicates of the same set of conditions.
I think I get it.
Yeah.
I think
Okay. Maybe we can
I
just
kind of
call
get it.
it four replicates.
Mhm.
Yeah.
A1 and A3 are A1, A3, B1, B3 are all replicates
All
of
the
the.
same. Yeah.
Okay. But these are for wild type and the others are for
Yeah.
mutant.
Yeah.
Okay. I think I understand now. Thank you.
Hopefully everybody gets that as well. Okay. Okay, veterinary medicine. Maybe. Maybe, yeah, veterinary medicines, veterinary doctors always have an idea about microbiology. Sometimes, if it's not your discipline, it's always. So what the thing is, I do a lot of systems microbiology as well. I conduct experiments in. So, for example, I can show you a plot. Yeah, something I'm working on. So this is like a combination experiment that contains about 2,225 compounds. I mean, no, 45 by 45, so that's 225 pairs of compounds, and they were all sampled in replicates. So I have biological replicates in there, I have technical replicates in there, I have replicates for the concentrations in there, I have replicates for the for the for the cells, that is the bacteria itself in there. I have replicates for the class of compounds as well. So it's a lot of, I'm used to this word of replicates. Uh, yeah. So I'm kind of used to this word of replicate. So sorry, pardon me if I was too technical in that place. But I hope every other thing makes sense from there on. Uh, yeah, you can rewatch the video, you'll be fine. That's Femi. Let's go to task 2.3. Who wants to lead the conversation? I'm honestly not a plant scientist, so I am having questions here. Yeah, knockout are the mutants. So the knock-ins should not actually be wild type, but in this case, we just call them wild type. We probably edit the question and correct it, but it's just white. Consider them as wild type. That's the knock-in. Anybody wants to lead the conversation for plant science? We need a plant scientist, really, or prop or botanist. Okay, maybe nobody. I'll try. So have a look at these data sets. Let's have a look. Okay, I'll probably reduce the size because it's difficult to read. Okay, this is actually a lot of data. Interesting. So some scientists are trying to engineer mutants for a crop. By the way, all the data sets you're having here, they're actually from publications. There's a publication generated, most of them. So, but we try to hide the publications. When we release because we don't want you to go and look for the publication. Because some of these publications also have the codes, and it becomes easy for you to solve. But when we are done, we give you the references and can probably get access to everything. So some scientists are trying to engineer mutants for a crop to become resistant to pesticide. So they compare the metabolic response of the engineered mutants to the metabolic response of the wild type. I think this is also similar to the previous one. So it's basically you have a mutant and a wild type, and you're exposing them to a treatment. So I think the geneticists can also cope with that. Basically, you have a mutant, you understand that as a geneticist, and you have a wild type. You expose them to something. And they took readings after 8 and 24 hours. So they took I think they did all metabolic profiling. Because you have many of these amino, not amino acid, metabolites. So guanosine, folic acid, succinic acid, blah, blah, blah. And your task is to calculate the difference in the metabolic response, that's delta M, between the DMSO treatment from the 24-hour treatment for the wild type and the mutant. Okay. Let's think about that. Does anybody have some ideas? So just as a disclaimer, I also don't have the solutions to everything yet, except the ones in microbiology and cancer. Okay, so this is DMSO. Actually don't recall exactly how the data was organized again, except I view it as a, is this a, what is the file format? Okay, TXT. I'll download it briefly. I just downloaded it briefly. Oh yeah, okay, it already exists on my pesticide treatment data. Okay, I will open with, let's open with numbers. Okay, it makes sense now. I was not getting the arrangement of the table. Okay, it's a lot. Okay, that's a lot. So you have the white type and you have the pesticide for the response of the white type to pesticide after 0 hours, the response of the white type to pesticide after 8 hours, and the response of the white type to pesticide after 24 hours. So in principle, I think what we were asked to do, what we were asked to do was to calculate the difference in the metabolic response, that's delta M, between DMSO treated and 24-hour treatment. So I would basically find the difference between these, the first row, and the fifth, no, 1, 2, 3, 4, and the fourth row. That's for the white type. Then I'll do the same for 6 and, no, wait, is it untreated or, hold on. Wait, between the DMSO treatment for, okay, yeah, I'm correct. So the DMSO treatment, this and the last one. So basically 6 and 9 and 2 and 5. So you calculate the differences between these two, and I guess you have to generate a plot, a scatter plot. Yeah, you have to generate a scatter plot for both. Now, it becomes tricky after that. You have to fit a line that satisfies a y-intercept of 0 and a slope of 1. Now, you are not fitting this line to the data set. No, you just want to put something that cuts through at exactly 45 degrees. I don't know if anybody understands that. Is that clear? It's very quiet. It's a lot of hot
Like,
science.
is it like best line of fit? Is it that kind of thing?
Yeah, that's the thing. It's not actually line of best fit. It's not related to the data at all. It's let's say, I think I personally included this. It's not really part of the task. So here is it. I can try to generate something. So let's say you have something like this. Let's say your data is like this. So if you are generating a line of best fit, it should go this way, right? But in this case, we don't want you to generate a line of best fit. We want you to generate something here that is exactly half. It has an intercept of 0 and it has a slope of 1. That is, when you have a slope of 1, the angle here is 45 degrees. That is, it proceeds in the same direction equally. If you have 1 here, you should have 1 here as well. If you have 1 here, you should have 1 here as well. I don't know if it makes sense to anybody. It's a little bit hard, I know. Sorry. Okay, now, using the residual cutoff, someone asked a question. Okay, it does. Using a residual cutoff of your choice. So by residual, oh, I shouldn't have deleted it. Yeah. So I would do some things. So using a residual cutoff of your choice, that is, residuals actually mean, did we try to explain residuals? But residuals mean the distance of any point to the line of best fit or to this fitted line. There is also a way you could derive that. It's part of what you have to discover, right? So it's like the vertical distance of that point to the line of best fit. One other thing I would say is if this becomes too difficult to do, you can also just use the line of best fit. Just use the line of best fit also. It's fine. But I think the line of best fit and this fitted line should be the same thing in the end. Anyways, when you now calculate your residuals is the distance. You can do this on R. We did not think, I don't think it's included. You can do this on R, you can do it on Python, sorry. And I don't think we included it in the course, but it's part of the things we want you to discover because it's a concept that is used a lot in sciences these days. So once you now know the residual, color your metabolites that fall within the range of the, okay, let's see. Let's say we have a line of best fit this way. Now everything that is, I'll replot it. I just replot it and clear all my drawings. Yeah. And then I'll look for an actual screen. Okay, I have something here. Let's say you have something like this. Then I want to plot this. So I've fitted my line of best fit, but then I want everything that is normal. That is everything that falls within your cutoff of whether you use plus or minus one. Let's assume you use plus or minus one. Oops. It falls here. They fall within this range. Yeah, so they fall within this range. Now, things that are outside, basically the outliers, that's what I mean, they fall outside, right? The things that fall outside, color them specially with another color. I think did I say choose? Okay, salmon. You can use red, you can use green, up to you. Just color them specially or color them separately. So color them separately. So it's like having a volcano plot, right? And you're plotting upregulated and downregulated after a specific cutoff. And you see everything outside this cutoff, they should be colored specially, right? Should be colored specially. I hope I'm not too technical. I'm scared with your silence. It's really loud. So now that you have these metabolites that are outside the range, what are these metabolites? How do you explain the trends you see on either direction of the plot? Pick any 6 metabolites that fall outside this range and generate a line plot that spans from 0 to 8 to 24 hours. Basically, let's say acetonitrile is what you picked. You can plot a line plot for these. Oops. That's my pen again. So you plot a line plot for this, for this, for this, and for this. So you should have something, if it is increasing, you should have something like this, this. Let's say this is your 0 hours. This is your 8 hours. This is your 24 hours. No, it shouldn't be. This is your 24 hours. Okay. So it's, and if it is probably decreasing, you should go probably the other way around, right? This was also found in a paper. So don't come for my head. It's something that people do. I will share the references later on. Actually, some papers have their references. Okay. Anyways, any question on that? We got specific requests for plant science. That's why we included it. Maybe nobody will do it. Okay, if there's no question, I move forward. I have 20 more minutes. Okay, biochemistry and oncology. So, any biochemists want to lead this discussion? Wow, guys are so scared. Should I call names? Nobody wants to lead the discussion again. Oops. Okay. I don't know why people are so scared. But let's say biochemistry and oncology. We want to discuss biochemistry and oncology. Okay. So protein structures are known to be strongly connected to their functions. However, at the amino acid level, not all amino acids contribute to the structure and function equally. Right? Now, Gagliardini and co., you could find a paper here, decided to investigate the impact of 4 possible individual non-synonymous blah, blah, blah. Basically, download the data sets, right? You look at the data set. Let's have a look. Okay. So if you have a look at the data set, it's quite a lot. Then you have your FoldX data set. So one of the data, I think the SIFT is a calculation of the function, and the FoldX is a calculation of the structural impact that an amino acid has on the entire structure. So it might affect structure and might not affect function. It might affect function and might not affect structure. But your task now is to actually verify if everything affects function and structure equally. So you have to import both SIFT and FoldX datasets, create a column for merge, and so you have to create a column, specific protein AA, which will be a concatenation. That is, you are basically, let's look at it. Is this zooming? No. Okay, so you have a protein and you have amino acids. So you have to create a column that merges these two information together on this place. This is FoldX and this is SIFT-X. Why not zooming? And this is, sorry, SIFT score. So it has the same annotation as well. So you can merge these two into a column, and in the end, we want you to have something like this. If you have A5, A607, and E63D, you should have a single value for the two of them as a separate column. I believe you guys can already do this. Now, using the specific protein amino acid column, merge the SIFT and the FoldX datasets into one final data frame. So this, I think it's easy for you guys. Now, a SIFT score below 0.05 is deleterious, and a FoldX score greater than 2 is deleterious. Now, using the criteria above, find all mutations that have a SIFT score below 0.05 and a FoldX score above 2. This is just if statements, right? I believe you guys also want to do this. If you don't know how to do it, please raise, if something is not clear, please ask. I know it's a lot of technical jargons that you have to know, but yeah, it just improves your competence with numbers. And it improves your experience across different data sets that you can play around with, different ideas on how to play around with data. Also, investigate the amino acid that has the most functional and structural impact. Okay. Will something come up? I also don't know. But it's up to you to decide. And hopefully So you can get the amino acid nomenclature here. Hopefully this link is still working. Oops. Okay. Okay. This one is different. It's a mutation, substitution mutation nomenclature. Yeah, but it helps you to understand, for example, if you have A5A607. No, sorry. If you have 5Y6, sorry, Y56C, it tells you that Y was substituted for C. And you should know, it's your job to also determine what Y is as an amino acid. So using the amino acid frequency table generated above, generate a bar plot and a pie chart to represent the frequency of the amino acids. So this is generating a frequency table should be easy for you. So basically it counts how many amino acids are, how many tyrosine do we have, how many, hold on. Okay, how many tyrosine do we have? How many valine do we have? And which of them is the most abundant in terms of function and structural importance? Then what can you say generally about structural property and functional property of amino acids with more than 100 occurrences? I'm also not a biochemist, so there's probably little I can also give to you beforehand, except I also go through the data myself. So yeah, please, I hope we understand. It's very quiet around here. Okay. Okay. Let's go to transcriptomics. So in transcriptomics, you have a processed RNA-seq data involving reads. Yeah, so basically you have RNA-seq data. Probably you already know RNA-seq, but RNA-seq is a scientific or experimental procedure where you sequence the RNA to quantify the number of RNA transcripts. So if you have a gene, if you say gene X, for example, how many transcripts of gene X exist in the cell or in the tissue at the time, right? So you have to generate a volcano plot from the data set. So volcano plot, you have to look for the meaning of a volcano plot. From there, you can generate a volcano plot. The volcano plots are almost the same everywhere. So, but you can check online and see what or how a volcano plot should look like. Now, determine the upregulated genes with log two fold change, that's log 2 FC, greater than 1, and those with p-value less than 0.01. Is it p-value? Yeah, okay, we use p-value in this case. And then determine the downregulated genes as well. So what are the functions of the top 5 upregulated genes and top 5 downregulated genes? I think this is one of the easiest ones. This one is just straightforward. After plotting, you split it into upregulation and downregulation. Yeah. So then we have something for the public health people. We also got special requests for this. That's why we included it during the registration period anyways. So this is a data set from the CDC. You can go to the data dictionary. It's like a metadata. So process the data sets, remove NA. This is a very hard step because R functions. Can you point to that? Maybe it's a mistake somewhere. But so basically everything R can do, Python can do it. At least for this task, for all the tasks. I can't see where the R function is, but if you can point me to it. So basically, you have to process the NA, sorry, either by deleting or converting to 0. Find out to do this. It's actually easy, but yeah. Visualize the distribution of BMI, weight in pounds, and age. So basically, you have to generate distribution plots. And of course, you can do this in R and you can do it in Python. What is the mean 60 seconds pulse rate for all the participants in the data? What is the range of values for diastolic blood pressure? What is the variance and the standard deviation for income? Visualize the relationship. So this is exploratory data analysis at best. And this is a lot of what they do in public health. So maybe you should find it interesting if you're a public health person. And then conduct t-test between the following variables, age and gender. Sorry, t-test between the following variables and make conclusion on the relationship between them based on p-values. So age and gender. So you have to also think about this because you can't really compare age and gender and submit it for p-value. Gender is male or female. I mean, it's categorical. Let's put it that way. And age is numerical. So you can't put gender into p-value. But you have to think of how to classify age into gender and gender into age, something like that. Then BMI values and the diabetes status, alcohol year and their relationship status. That is, how long have they been taking alcohol? So yeah, that is all very easy. Okay. Yeah, Kimat, you have to categorize. Yeah, you have to find a way to, basically it's categorization. All the ages that belong to female, you collect them on one side, and all the ages that belong to male, you categorize them on another side. So in this case, we specified accurately that you should conduct a t-test, but you can also think about it. Maybe there are better ways to do it. The introduction to the transcriptomic question. Okay, I saw it. We'll correct it. It shouldn't be base R functions. It's explore the data, basically. That should be it. By base R function, we just mean don't import any library. Like just use the way R is, use it that way. Yeah. So it's just base R, but in principle, what base R means is that basic R, don't use any special thing, don't import any libraries. But I think for the people in Python, you are allowed to import libraries. You're allowed to. You can import your pandas, you can import your matplotlib or your Seaborn, up to you. Okay, and that will be all. I think I was able to answer most of the questions I got. Otherwise, Yinka was unable to join us anymore. I think he joined late, and I was asking for someone to help. He didn't raise up his hand. Okay, thanks to everyone who contributed. If you are still a little bit shaken by the questions, feel free to ask questions. Watch the recordings again. Hopefully they could help. And good luck with the coding. Yeah, bye everyone.